Hi, I'm Matt Scholz with the Sustainable Phosphorus Alliance, and thank you for joining us for Phosphorus Science Now. When we think of sources of greenhouse gas emissions, we might be more inclined to think of smokestacks and tailpipes than lakes. However, lakes are major sources of emissions that are now thought to account for perhaps 12 to 23% of the CO2 equivalents of fossil fuel emissions. Most of this impact is thought to be from methane emissions, which occur naturally and are rising, in part because of phosphorus-driven eutrophication. We've known for a long time that lakes emit methane when bacteria at lake bottom break down biomass in oxygen-depleted environments. But only recently have we begun to appreciate that areas nearer to the surface, where oxygen is much more abundant, release significant amounts of methane. While the source of these emissions is still under study, the prevailing hypothesis seems to be that certain bacteria in these subsurface waters metabolize a group of compounds called phosphonates, and in doing so release methane as a byproduct. They do this, interestingly, in waters that are otherwise phosphorus limited. Phosphonates are compounds that contain carbon-phosphorus bonds, and they are both naturally occurring and synthetic. Among the synthetic sources, glyphosate, the active ingredient in the herbicide Roundup, is a major source. Recently, a Japanese group published a detailed analysis of how subsurface methane is produced in deep freshwater lakes and studied the relationships of carbon, nitrogen, phosphorus, and other factors with its production. The lead author of that study, Santona Khatun, a graduate student at the University of Yamanashi in Japan, joined us for Phosphorus Science Now. Santona Khatun, thank you so much for joining us for Phosphorus Science Now.
Thank you, Matthias Scholz, for giving me this opportunity to present my study today. Thank you.
You're very welcome. Could you tell me who your collaborators were for this project?
So the first one is Dr. Tomoya Iwata. He was my supervisor from the University of Yamanashi. And the other collaborators is Dr. Hisaya Kojima, Dr. Tanaka Kenta, Dr. Ryuichiro Shinohara, Dr. Hiromi Saito. And the other three is my laboratory members, Yoshiki Karashi, Kana Yamanami, Daichi Mazawa.
Okay, great. You have a big team. So
Yeah.
can you tell me a little bit about the research that you did?
It's about the aerobic methane production in the freshwater lakes. Previously, we have known that methane is mainly produced in the subsurface, in the sediment of the lake. However, recently now we found that the methane is also produced in the oxic layer, which is mainly by the microbial microbes in the freshwater lakes or in other ecosystem. So based on those previous study, we also wanted to perform or know the unknown part of this aerobic methane formation in the freshwater lakes.
And you use the term SMM a lot in this presentation, I think, and so that's the subsurface
methane maximum.
Methane maximum. Okay, great.
If I want to define this subsurface methane maximum, so it is like in the layer, especially in the metalimnion of the lakes, we found the high peak of methane, even from the epilimnion or in the hypolimnion of the lakes, which is we defined as the subsurface methane maximum. Especially in the marine and in the freshwater lakes, we found that the subsurface methane maximum as well as the aerobic methane production was observed. And in the list, you can see the microbes and name of the substrate and as well as the references. These microbes in the marine ecosystem utilize several phosphonates and thereby producing methane, especially in the oxic layer of the ecosystem, marine ecosystem. In case of the freshwater ecosystem, we also found that several planktonic bacteria, Pseudomonas or Synechococcus, also utilized different substrates such as trimethylamine or phosphonate, thereby producing methane in the oxic layer. So in fact, this is Khatun et al. 2019 is my previous study where we hypothesized that, where we show that the Synechococcus having CP can cleave the carbon phosphorus bond and thereby producing methane in the oxic layer. And based on such study, like most of those studies are about the how, only about the substrate and the microbes. However, about their physiological environment or other factors was not known yet. Therefore, to show how the lake physiological environments linked with the microbial community and the SMM formation, I performed this current study. As you see here, this is the objectives: to clarify the pattern and mechanism of the SMM formation, or to identify the variable, how the variable affect the SMM formation, or the microorganism responsible for the aerobic methane production. This is the geographical in the left side. This is the geographical map of Japan, and the lakes are known as like 1, 2, 3, 4. You can see the Lake Ashino, Lake Motosu, or Lake Saiko, Lake Aoki, Lake Nojiri, Chuzenji, Lake Hibara, Lake Tua, and Lake Shikotsu. These are the 9 lakes.
Great. And can you tell me what were the conditions of these lakes? Are these all very pristine lakes? Do they have high phosphorus loads, low phosphorus loads, high nitrogen, low nitrogen?
Most of the lakes, the average total phosphorus concentration was 0.03 to 0.3 micromolar, so which is very low phosphorus
Quite low,
condition.
yeah.
Yes, in case of the nitrogen also, they had 17 to 33 micromolar total nitrogen concentration, so which is also could be considered as low. So most of the lakes are mesotrophic, oligotrophic, or ultra-oligotrophic in condition. So we performed the field survey for this lake study in between 2016 to 2017, with the help of Dr. Toma Iota and other our laboratory members. And several, especially we collected the sample at the deepest point of the lake, and using the 10-meter depth, like 10 depth for each lakes. And the environmental and the chemical and biological parameter were also analyzed. Especially the phosphonic compounds, which was analyzed through the NMR analysis, was performed by Dr. Ryojiro Shinohara, and the community structure of bacteria, which was performed by Dr. Hisaya Kojima. This is the vertical methane profile for each lakes. So you can see here is the 9 graph. So 9 graphs indicates the 9 lakes, and where the vertical profile indicates the depth of each lakes, and the upper horizontal axis, which indicates the dissolved methane concentration, and the lower horizontal axis indicates the dissolved oxygen concentration. And here the red circle indicates the methane concentration, and the gray circle indicates the dissolved oxygen concentration. And if you see most of the lakes here, this shaded area indicates the thermocline of the lake. Most of the, in case of the lakes, the subsurface methane maximum was observed in the near the thermocline of the lakes. So we, this in first analysis, this was our result, that the SMM was formed near the metalimnion of the lakes. And secondly, the question arising, what is the potential factor for such SMM formation? In my study here, we observed that the SMM could be formed by the dissolution of atmospheric methane, or the tributary river using the lateral transport of dissolved methane, or the methane transportation from the littoral zone, or from the hypolimnetic methane from the deepest point. However, this is just a schematic diagram for Lake Saiko, one of the study lakes. However, we found that in Lake Saiko, where the methane profile, you can see the methane profile, and which was closely associated with the dissolved oxygen profile. As we found this kind of phenomena, so we can say that the lake microbial community might be related to this methane formation through different biogeochemical processes. Then question arising, what are the biogeochemical or variable affecting the dissolved methane concentration? Therefore, to identify the biogeochemical processes, I select here 9 variables. So you can see the depth, temperature, surface irradiance or chlorophyll A, DIN, SRP, DOC, DOP or TNTP ratio, which are the 9 environmental variables we selected and performed the generalized linear model for those. And from according to this generalized linear model, we found that 6 variables using here you can see the regression line for the 6 variables which were found by the best model, and where the sum of the achic weight revealed that the DIN concentration, like DIN was the most important variable that affecting the dissolved methane concentration. After this analysis, when we found that the DIN or DOP or TNTP, which showed the important relationship with the dissolved methane concentration, we wanted to know what is the variable which impacts on this chemical variable. To identify the factors that affecting the chemical variables, we did the further generalized linear model analysis, and we found that the DOC concentration, which was negatively correlated with DIN, similar to the previous slide, as you see here, DIN was also negatively correlated with methane, and the DOP, which was significantly positively correlated with DOC, and the TNTP was negatively correlated with the DOC concentration. So based on this result, we can say that the high DOC and low DIN relationship might impact on the microbial community and subsequently control the methane production. And then we wanted to know the microorganism that relating to the methane concentration, and this analysis was performed for 3 lake samples using from the epilimnion, metalimnion, and the hypolimnion. And then we performed the community analysis for these 3 layers, and these are the result. Relative abundance for the left-hand side figure indicates the relative abundance of the microbial community, while the right-hand side, which indicates the relationship between the microbial community and the different layers, I mean these 3 layers. So this NMD and DS analysis, which revealed that here you can see this Cyanobacteria, which was closely associated with the methane gradient, although we could not find very significant relationship, but we found that the Cyanobacteria, which was present in the epilimnion, were related to the methane concentration. And then for further analysis for lake methane and Cyanobacteria, we again did the several analyses for microbes such as chlorophyll A or different bacteria, eubacteria or type 1 methane oxidizing bacteria, Cyanobacteria, Synechococcus, Archaea or algae. And here the vertical axis indicates the methane concentration and the horizontal axis the each microbes. And we found the significant relationship was only observed for Cyanobacteria and Synechococcus. From this analysis, we again confirmed that maybe Cyanobacteria or Synechococcus is related to the dissolved methane concentration. And then to clarify what is the relationship between the vertical methane profile and the Synechococcus, here is the result. And again, likewise, the vertical methane profile, we did the same analysis for the microbial community and for the 9 lakes. And here the vertical axis again indicates the depth of each lakes. The upper horizontal axis indicates the Synechococcus cell density, and the lower horizontal axis indicates the dissolved methane concentration. And blue and red circle respectively indicates methane and Synechococcus. Where you can see in the shaded area where the methane production was observed, Synechococcus cell density also increased. And based on this result, we again can say that Synechococcus cell density is closely associated with the dissolved methane concentration. So for further analysis between the peak methane concentration and the environmental variable, we did the further correlation analysis between them. And here, I first want to introduce the peak SMM, which is the highest concentration of supersaturated methane. And when we perform the analysis here, the vertical axis indicates the peak SMM, and the horizontal axis indicates the each environmental variable. And you can see that the positive correlation was observed for DOC, DOP, and Synechococcus, while the negative correlation was observed to the DIN and GNTP ratio. So based on this analysis, again, we here wanted to know what is the ratio between the DIN and DOC and the microbial and methane concentration. So when we performed the analysis between this DIN and DOC stoichiometric ratio, we found that Synechococcus and methane both had the decreasing negative relationship. Based on such relationship, we can say that the Synechococcus might regulate the methane concentration as well as the subsurface methane formation in the freshwater lakes. As the Synechococcus has the CP-lyase ability, so we also hypothesize that maybe the Synechococcus can decompose the dissolved organic compound in the lake and therefore producing methane. So to identify the presence of phosphonates, we again performed the NMR analysis, and interestingly, we found the phosphonate compounds in Lake Toya, not in other lakes among our study lakes.
Okay, so can I ask you, so Lake Toya was the only lake that you found phosphonates in, is that correct?
Yeah, I only found in Lake Toya. And based on this presence of the phosphonates, although we could not find it for the other lakes, we can summarize that maybe the Synechococcus are the organisms. So here I want to make a summary for this Synechococcus, which might have the CP-lyase enzyme that could use the DOP phosphonates and thereby producing methane in the freshwater ecosystem. In the previous study, DOC are also known to influence the methane emission of the lake ecosystem. However, here we found that DOC in the water column is responsible for the methane formation. So we wanted to say that. So maybe the DOC loading under the climate change could impact the end depletion, may cause the end depletion to the Cyanobacterial domination, and therefore help the cyanobacteria to utilize the phosphonates and thereby methane production in the oxic layer.
Okay, great. That was a very nice presentation, and I thank you for joining us.